Our next speaker is Professor Rick Wagner, professor and endowed chair within the Department of Medicinal Chemistry at the University of Minnesota. He's also the director of the Chemical Biological Initiative in the university's College of Pharmacy. Professor Wagner completed his PhD at Duke University and was an NIH postdoctoral fellow in the Department of Chemistry at Penn State. His research group in Minnesota seeks to apply the principles of organic chemistry, enzymology, analytical chemistry, molecular and cellular biology, biophysics, and nanotechnology to the development of new methods for drug design and delivery, chemically engineered cellular therapies, and tissue engineering. Today, he'll be speaking to us about his work in engineering cell-cell interactions with chemically self-assembled nanorings.
Great.
Okay, Professor Wagner.
Yeah, let me get my slides up. Let's see, can you see that? Let's see, I go to.
Yes, as soon as you put it in presenter, there we go.
Is it in the right order, or do I need to switch it? I need to switch it.
Looks fine.
Okay, great. So I want to thank James for the invitation. Sorry, it kind of got back and forth in email. My horror is actually missing emails, and then like they just get piled up, and so kind of going back and forth and things. So I'm glad we finally were able to connect. I first want to thank. I want to say how much that characterization lab, how much I think it's fantastic, such a fantastic thing. And I loved your talk today earlier because I learned some more things and things we might be able to take advantage of. We're actually moving into microfluidics, and so that was. I think we've already talked to you guys over there about helping us with that. So I really am grateful for the existence of that facility. Today, what I'd like to talk to you today about is a little bit different. There's going to be more biology here, and so if you do have questions, please please ask them. That I look forward to those questions. But what my lab has been interested in is how we might be able to use nanotechnology for the engineering of cell-cell interactions, and this all came out of building a material that we didn't really know what we're going to use it for, and then that evolving into how we might think about how we might ask questions about cell-cell interactions. Now, the engineering of cell-cell interactions is actually really an important biomedical field these days. It's been in all the sort of papers, and people know all about immuno-oncology and the huge growth in immuno-oncology. And one of the most important areas in immuno-oncology is the actual design. Let me just get my pointer here. Is the actual design of cell-cell interactions genetically, in which this is a CAR T cell. It's referred to as a chimeric antigen receptor T cell, in which a T cell from a patient is engineered to contain a antibody, a binding part of an antibody that can bind to a tumor cell through a recognized ligand, and then it's fused to a activating domain that will activate the T cell once it's interacted with the tumor cell. Now, like I say, there are actually products that we have on the market now. This is the first. These are truly individualized medicines, because a patient's T cells have to be engineered, and then are given back to the patient. And the tumor is attacked by the T cells. Right now, this is entirely towards the sort of lowest hanging fruit, which is B leukemia applications, and it's been fantastic. There have been a number of issues, though. One is from an engineering standpoint. The actual chain, the actual product making these cells and point of care is kind of a nightmare, actually, because you have to be able to make it from the patient's cells, and they have to be delivered at a certain time, and so they can't be delivered immediately. They have to be given over the cells. It takes either 4 to 6 weeks to actually prepare those cells. In some cases, patients have died before the therapy has been allowed. They've gotten the therapy. It's not particularly still efficient. You still get, 40 to 60% of the cells are actually effective. You can have long-lived chronic damage to normal tissues, particularly if this antigen is on normal tissues. So that's one of the potential side effects. And the manufacturing costs are very high, and you can't really turn it off. So once your cells are, you have those cells, they can be with you for life, and therefore you have to deal with them for life. So, and when I mean manufacturing costs are high, one injection of these cells costs upwards of $500,000, and a patient for the full therapy, because the patient has to be hospitalized during most of the time before they get this therapy, the patient actually costs, or plus the therapy, is upwards of 1.2 million. So there's a lot of what's been exciting. There's a lot of issues here with regard to using this therapy. Now, there have been a number of ways of potentially thinking about non-genetically engineering cells, ways in which you would be off-the-shelf ways that we could do that. We could engineer cells, cell-cell interactions. And I'm not going to go through all of these, but there's a number of ways using chemistry, bioorthogonal chemistry, very clever ways that have been developed. However, none of these methods are really very translational. In other words, that means is that it's very hard to get these potentially into the clinic and potentially used as medicines. So a number of years ago now, a bit ago, we were looking at how we could design protein-protein interactions. And one of the molecules that we were working with was this molecule, the hydropolic reductase, and it binds to a ligand called methotrexate. If we fuse one DHFR to another DHFR and then fuse it to a targeting ligand and then mix in a bivalent ligand, what we found is that we got these to actually self-assembled into nanorings. And we can actually tune the size of these nanorings based on the linker length and composition between these two DHFRs. So this is all one protein, it's a monomer, and this is a chemical. We mix them and we get the nanorings, and here's some AFM to show you to show you some of their composition. Now this gives us a scaffold. We recognize that this was a pretty stiff molecule, and it's about 35, somewhere around 35 about 20 nanometers in size, but it's almost like a washer because it's only about 5 to 6 nanometers in height. And oh, I think something's out of place. Sorry about that. Let me just, sorry. So we made some of these. This is with a molecule that's targeting CD3, which is on T cells. And we found that because they're multivalent, you can get avidity effects in which if you have the monomer here, say this is a monomer CD3, as a dissociation costs about 50 nanomolar on cells that are expressing the antigen, that if we make the octamer, we can get an enhanced binding affinity to those cells. Now, after a while, after studying these for other uses as well, we began to think about if we were able to take two monomers, one that would target, say, the T cells, and one that would target a cancer cell, and our bismethyltrexate, and we mix them, we would get actually, because it's an octamer, stochastically, we would get 99.9% basically bispecific. Now it's a stochastic mixture, but it's very heavily weighted for 3 to 4, 4 to 4, 4 to 3, and so forth. That actually has turned out has been actually useful because there's some heterogeneity actually in the mixture, because cell surfaces are not homogeneous, they're heterogeneous. And so this allows us to make rings that are bispecific, that can able to bind to T cells and bind very tightly because you have high avidity, and then allow them to then target to potentially a cancer cell, activate the T cell, and then potentially kill it. Now, in this way, we're making basically rings that will bridge basically that T cell. We bridge the T cell with the cancer cell, And we refer to these as prosthetic antigen receptors because what we've done is we've put a prosthesis on the T cell. So the T cell has got basically a prosthetic receptor, but it's because of the potential long life of this, it's going to act like it was genetically engineered. So we can make these are proteins, and this is an SEC, and we can make the two monomers. We can mix them. You get this typically large for the octamer. You get this large peak for SEC, showing you get a much larger species. When we do DLS analysis of these, we get a nice, clean, nice release, nice peak with a rate of hydration of around 16, but very, pretty narrow. If we do cryo-TEM, we've done cryo-TEM analysis of these, and that's sort of our standard now. And what we can see is we get a nice distribution that pretty much centers around the octamer, and that it corresponds to what we would see in the DLS. Now, if we look at how they might bind to so they can bind to T cells or to the cancer cells. And here we're using an antigen EbCam, which is on a lot of endothelial cancers. It's actually on almost all of them, in fact, and has been used as a marker for many years. What we can see is that if we make the bispecific rings, they will bind, and this is a flow cytometry and FACS analysis, and what we can see here, this is an increase in fluorescence. So, and this is the number of cells. And so as we increase in fluorescence, it means we have more rings, we have more binding to the cells. And so we can see that the bispecific rings bind to the breast cancer cells, and they also bind to the T cells. Whereas the individual rings, the rings themselves that just only bind to T cells or bind to EbCam, don't bind either one of those cells. And we can see here, then, here's the breast cancer cells. If we then add, this is confocal microscopy, if we add labeled bispecific rings to them, you can see it's binding. They've been labeled green, and they bind all the places that you would expect EbCam to be expressed on these junctions. Now, a key point is that we said is that if they have a high avidity, then they should have high stability on the surface of the cell. It won't do us any good in vivo if they are just if their stability is low. And what we found is in this competition experiment that when you add the cells and then you test this over days, that you have greater than 3 days stability on the surface of the cell. So they really stick well and they stay on that surface of the cell. As the cells start dividing with time, they slowly divide, you start to lose some because, of course, they're dividing and you're getting less on the cell. Now, a really key feature of what we can do is we can use a drug called trimethoprim, an FDA-approved drug. And this drug, you've all taken it, particularly if you've had children who've gotten sinus infections or any sort of those childhood strep infections. It's very common, and you've gotten it too. Trimethoprim is used. It's almost 100% orally bioavailable, and we can add trimethoprim, and what that does to the cells or to the surface is remove basically the rings from the surface of the cell. So we have an ability to turn the cells on and turn the cells off, and so this gives us a switch that gives us potentially a safety switch, actually, that we could potentially use. Now we made the rings, and I'm going to just show you some live cells here. Hopefully this works. And these are this is a live cell imaging, and what you see here in the red, this red is are glioma cells that have been stained red, and then these breast cancer cells up here are the smaller cells. Now those cells are this is a mixed culture, and the breast cancer cells have the antigen, the glioma cells do not. These little guys wiggling around, those are the T cells. They're lurking and searching to see what cells they might recognize and to be able to kill them. Now, nothing's really happening in there because they haven't been modified. Nothing is happening. So if we modify the cells, and I might ask you to look down here at this bottom part, you'll see that the cells are starting to collapse and die. There are lots of T cells that are around them, and they're basically killing them very quickly. Whereas the glioma cells are not touched, and basically you have all of this sort of garbage that's from the breast cancer cells, and the T cells go on to kill more of those cells. So we went into decided to go into animals now, and so in this case, what we did is an orthotopic model. These are animals which have no immune system to speak of, and we're going to give them human T cells. We can implant them, and they'll grow out within the animal, and the animal will have basically human T cells. And then we're going to give the breast cancer in the mammary fat pad. So orthotopic means that it's going to be placed in the tissue that you would find the breast cancer, which is in the mammary fat pad. And in this case, and we'll visualize them, and we chose to label the cells first, just like with CAR T cells, and then periodically, either a day, 2 days, 4 days, or 8 days, we would inject then more rings to rearm those cells, because it was clear to us that over a period of time, as the cells divide, they lose their potency because you're dividing the amount of rings that are on that surface. And so you can rearm them by adding more rings. And what we found is that this is the injection of the cells, and then this is our controls that we have that are just the T cells, just the non-targeting rings. And then and we gave this, in this case, we're giving the injections every 2 days, up to a maximum of basically 6 injections or 5 extra injections. We found very quickly the second injection, basically these tumors were being obliterated, and essentially the tumors in the animal were gone. They're eradicated from the animal. Whereas all the other controls had tumors just completely grow out to be extreme, very large. And so these animals were essentially cured from it. Now we looked also at the dosage. How many could we get away with? What's the number of doses that we could get away from, or spacing them? What we found is that these are curves in which we gave it every day for 5 days, every other day, another 5 for every other day, every 4th day. So 4th day, and we could still see tumor eradication over the course of this experiment. And then even if we go out to every 8 days, we still see a pretty good, it's only given once every 8 days, we still get a pretty good tumor suppression. And so this allows us to think about clinically how they can be used, because it turns out patients don't want to be taking, if you talk to clinicians, they don't like taking drugs every day or every other day. They would like to come in once a week to do it, or once every 3 to 4 days tops. And so you need to think about your therapy, how your therapy is going to be implemented, and how well the patients will meets their potentially needs. So this gives us an option that we have within that 3 to 4 day window that gives us some information about that. Now, there is no toxicity. This is weight over the period, and there's essentially no we don't see any toxicity from this, from the doses. The animals all were fine, except those animals which started to have tumors grow out, the controls. Now, if we look at the animals, one of the key things is how long do those T cells stay on the cells, and how stable are they? And also how fast do they get into the tumor, and how well do they start beginning what they need to do? And so if we look at this middle part, let's get my laser pointer. If you look at this middle graph, what we found is that once we inject the rings, when we inject those rings, that the rings are in the tumor. Actually, quite a bit of the rings are in the tumor, labeling T cells, and that are actually, there's T cells already in that tumor, T cells and the tumor itself. Within 6 hours, it basically maxes off. So very quick. We can block that with an antibody that basically binds to the same antigen, and we can block that interaction. And just rings that are not targeted don't do a very good job of binding, of being located in the tumor. Now, if we look at if this is a biodistribution, this is the tissues in which you would find these rings. Now, the way we did this, I want to point out, is that the bismethyltrexate can be radiolabeled, and that's what we did. And so if the rings come apart, this bivalent ligand will come apart too and is rapidly eliminated. And so we can always tell if the rings are intact, because in order for them to be radiolabeled, the protein's not labeled, but the chemical dimerizer is. And so as a consequence, what we see is that the rings themselves are where you would find T cells in the animal. We find lots of T cells in the spleen, in the liver, There's some in the kidneys. Basically, you see some elimination in the kidneys. There's some in the testes, and then in the intestines. And then if you look at the tumor, you can see a much larger amount in the tumor, relative to the non-targeted ones. If we look over here, if we look at the T cells, if we look at the T cells in circulation, what we find is within 6 hours, those T cells are labeled, and then the circulating T cells are all labeled for upwards of actually 2 days. And so that means we have a very stable modification of those T cells. Now, what about the trimethoprim? Can we use that to actually knock them off the cell, knock them off the T cells in vivo? Because that's what we'd want to do in the clinic. And so what we find here, we're looking at the markers for this are cytokines. These are very, very reactive molecules that are produced in an inflammatory response. In fact, for coronavirus, one of the bad things about coronavirus is for some people, and we don't know why this is the case, is the elevation of the inflammatory response, and so until it gets out of actually control. And a number of the antigens that are the cytokines that get upregulated that lead to activation of the immune system are interferon gamma, IL-6, TNF alpha, and IL-2. And so what we find is in this experiment, the animals have their tumor, they've been given their T cells, they've been given basically the rings, they've been given a dose of the rings, and you can see their cytokines are going up. This is a good thing because it means that the T cells are immediately within 1 day recognizing the tumor and starting to kill it and getting activated. If we give, past that 1 day, a dose by IV of trimethoprim, just one single dose, and the trimethoprim has no toxicity to speak of. What we can see is that, and look out 24 hours later, we can see a drop in those cytokines, and these are the things that we want to, so that we're deactivating those cells. That's important because the main toxicity for immunotherapies, particularly CAR T cells, is overactivation of the immune response. And so what you want to be able to do is have an antidote to that, be able to knock them off. And it looks like we can use an FDA-approved drug to do that. Now, if we look at T cells, I don't expect you to know, this is sort of for the T cell experts, but I want to just point out that we have there's a number of different types of T cells that are important here. The T effector cells are the ones that actually kill the tumor cells. There are memory cells you carry in your circulation, memory T cells. This is part of what you get when you become immune to something. These effector memory and central memory cells are the memory cells that remember, so that the next time you get sick, they can rapidly come online. And what we see here is that our effector memory cell population is expanding for both CD4 and CD8 cells. These are two different types of CD4 cells. The CD8 cells are the ones that are actually the main killers, and the CD4 cells help this immune activity to keep going, to get upregulated. And then these are the effector cells are really expanded. So we see an active response across that time that engages the tumor. Now we tried this on another antigen. So EbCan works. What about another antigen? So we looked at an antigen that actually is for cancer stem cells as well as on primary cells for some tumors. And this is a colon cancer cell line. And so in this tumor model, this is the same model where we have NSG model mouse, has human T cells, and then it has implanted into it basically a human colon cancer. And what you can say here, again, we see the same effect. Within 5 doses every 2 days, we can get eradication of the tumors over time. And then also when we look at this it's called a survival curve, Kaplan-Plotz curve. We see we can get 80% of the animals are actually cured. One of the animals developed a tumor kind of late. But this gives us, and this is where our controls are. So it looks like it's really positive. It works for, now it's our second antigen, and we've also done a couple of others. Now, that's all well and good, but tumors are quite heterogeneous, and one of the issues is how you get a thing called cancer or tumor stem cell-like cells. the hypothesis has been that when you have a tumor, you have primary cells and you have these stem cells. These stem cells, once you hit a patient with chemotherapy, they're fairly resistant to chemotherapy and radiation. So you pretty much can wipe out the primary tumor, but the residual disease that develops later while your person comes out of remission is due to these stem cells, and they start to grow out and differentiate into the primary cells. Now, if we hit the stem cells, we were able to hit the stem cells, then the theory is that you could go into you would be left with just the primary tumor cells, and you would have you get tumor remission. However, what's been noticed in the last few years is that even if you hit the stem cells and you can potentially get rid of them, what happens is you can get phenotypic switch. What that means is reversal, is that primary cells, if they sense that there's no stem cells there, can potentially de-differentiate into or revert into these stem cells and leading to tumor outgrowth. And this is pretty scary, actually, because it means that you really have to think about two different cell types of populations and not look at a tumor as just some sort of homogeneous set of cells. And so we were wanting to examine this, and this breast cancer cell line 231, we realized, has EBCAM is our primary tumor model, and it also expresses some cells express CD133 positive, and these are CD133 positive. So these cells would be where you would get the tumor initiating cancer stem cells, and they're a subpopulation of this cell line. Whereas most of the cells, 90% of the cells, are actually these primary cells. Now, how would we address this? Well, what we could do is we could use our EBCAM rings, and if we mix them with our CD133 rings, then what we'd be able to do is we would be able to hit the three different populations. If we had just CD133, we'd only hit two of the possible populations. If we had just EBCAM only rings, we'd hit only two of these populations. But if we were able to outfit the T cells with both rings simultaneously by just simply mixing them, we'd be able to hit all three of those populations. So again, the CD133, as I showed you, works pretty well. This is cell lysis. It kills the tumor cells quite well at a nanomolar range. And then if we take our 231 cells, what we see is that we only get maximum killing when we actually have a one-to-one mixture. We have a mixture of both of those rings that can kill both types of cells that are in the cell culture. So we went into animals again. Now in this case, we're going to do orthotopic model. That's going to be a 231 cells that are 231 cells, which is a triple negative breast cancer cell line, which is very hard to treat. We're going to do a model again now where we implant the tumor cells, the T cells, and then we're going to give the drug, and then we're going to give the ring. Look at what the effect of the rings would be. So if we look at controls, so here's the tumor volume. Here's over the days. Our controls, the tumors grow quite well. Just the PBMCs or non-targeted rings. We're going to give like 6 doses. If we target just the EBCAM, all right, the primary tumor, we get a nice tumor decrease, but we never get down to 100% loss of the tumor. And after we've stopped, the tumor actually starts to grow back again. If we hit with CD133 only, we can actually block completely tumor growth. It stops. It basically gets stabilized. We can't get rid of the tumor. The primary tumor cells are still there, but it's these apparently these stem cells that are really important for tumor proliferation. Once we stop this, we actually can start to see this tumor start to grow again, indicating that when you get rid of those tumors, those T cells, those stem cells, there may be plasticity in the tumor, so that some of those tumor cells are actually reverting back to stem cells. When we hit them with both, we can essentially eradicate the tumor. And we don't see any outgrowth afterwards. And so hitting the stem cells and hitting the primary tumor cells appears to be critical to eradicating this heterogeneous tumor. If we play this out, what we see is that actually with the two single ones is that the actual kinetics of the tumor growth becomes actually quite, they actually match each other. And so these have become essentially the same. And in fact, we get long-term tumor suppression. One of our animals started to develop late a tumor. And what we play this out, we can see over time to the end of the experiment is 80% of the animals had their tumors eradicated and were cured. If we hit just the stem cells, it actually is great because the stem cells apparently matter more than hitting the primary tumor in terms of the longevity. This is a waterfall, so each one of these drops is a loss of an animal in the study. So again, in this case, we don't see any toxicities in this case. And so we wanted to investigate this a little bit more. And in this case, what we've got we basically deplete now the tumor of all these CD133 cells, the tumor cells in vitro. And this is again, a FACS analysis in which this is fluorescently labeled cells. And what surprised us is that it didn't surprise us because of the in vivo, but is that what happens is that so you can go from the sort of what you started with down to 0, and then within a week those CD133 cells are coming back, and so within a week you're starting to see reversion of cells back into the back to this normal to the stem cells. When we do it in an animal, we actually can deplete the CD133s, and this really shows you the tumor is really slow. Its growth is greatly decreased, but eventually over time it really picks up steam and starts to grow. If we add in more CD133 cells, it actually even increases the rate of growth of those tumors. And then if we look at using FACS analysis, if we look at the number, this corresponds, this 18, 17, and 20 corresponds to the number of CD133 cells at the end in those tumors at the end of the experiment. They're essentially the same. So what does this mean? Not only do we have plasticity, these primary tumors are going back to stem cells, but then they're reaching some sort of homeostatic level. In other words, they don't just swamp out the tumor. They actually there's like some signal that's telling them to only go to a certain number of cells or percentage of those cells. And this is all quite fascinating. We're trying to figure out what this is telling, using these tools now, telling us about how tumors grow and maintain themselves. Now, these are primary. I forgot to say, it was in my first slide, is that this technology has been licensed to a company that I've started up called Tycon Biosciences, of which I am the CSO. And we've been looking at how we can translate this. And one of the most important things is immunogenicity, because these are proteins, foreign proteins. And what we see is that this is the level of antibody that's formed when we inject these rings into animals over a period of a month. When they have a full immune system, is we see almost no production of antibodies to the rings, and we see no basically activation. This IL-6 is low, of just splenocytes or B cells and things. And so right now what we've been doing is within the company is further humanizing the CSANs, and they're almost completely humanized. So we can start planning how preparing this for the clinic. Now we've looked at how we can use these in a practical way, and how we can use these nanorings for a T cell response. We thought about we have to have, in that case, an antigen on both cells. Is there a way to think about this to make it a little bit more universal? And so we thought about, this is the bismethyl-trexate, and then we have a version linker that has a third arm. So this is responsible for the self-assembly. And we've been able to use that to conjugate to a variety of different things, like oligonucleotides, radiochelators, as I showed you before, drug molecules, fluorophores. And so that's all been useful. What we thought was, could we use this to help us in designing rings that would be potentially able to bind to membrane surfaces? And so we constructed a trilinker which has this azid on it, which has a long peg linker, which gives us a water solubility, and then has this azid on it. And that azid is so we could potentially use click chemistry to be able to click reactions onto the surface of cells. And so here you see, if we have, for example, we thought if we have lipids that have a reactive moiety called DPCO, and then if we make a ring, which the trilinker has this azid on it, these would click form, do the click chemistry on the ring and bind to the surface of the cell. And this would allow us to then essentially uniquely modify that cell and then allow it to drive its cell-cell interactions with another cell, which would be EbCAM positive. And then we could use trimethoprim to break this up, to disassemble these, engage, so we could have control over assembly and disassembly. It also occurred to us that we could use another old favorite, which is biotin streptavidin. In this case, this would be monostreptavidin, so it's just, streptavidin has 4 binding sites. In this case, it's a version in which we only have 1 binding site. And so it's not as high affinity, but because we have avidity effects, we can get very high affinity to the we propose to the surface. In this case, we're going to use a lipid that has biotin on it, that displays biotin. And this would allow us to now have a non-covalent interaction in which we add these, and it would bind to the biotin in the membrane surface, again driving cell-cell interactions, which then could be disrupted, disassembled by the addition of trimethoprim. So this is to show you that it turns out that neither one of those lipids affect cell viability. You can take it up to high concentrations. You don't see any death of the cells. You can also increase the concentration across the cell lines, and in this case, of course, there's a marker where we test the amount of lipid that's there fluorescently, and we can, as we increase either in these MCF7 cells, breast cancer cells, or B-cell leukemia cells, we can see as we go up in the concentration of the lipids, we get more and more of the lipid, inserting into the membrane. This is true for the biotin as well as the DPCO-based ligand. So we can think about to install these, we could have the MS. We looked at if you just had the streptavidin rings, displaying rings, and you can see they strongly bind to the cells at a very sharp peak. This is really nice. We have increased fluorescence. If we do the click chemistry, again, the rings bind nicely. The chemistry goes on, and we get a nice increase in the fluorescence. And then, of course, if we have just a binder that binds to a surface protein on the surface, we get surface binding. So there's like 3 different ways of binding to that surface. And of course, we can make a bispecific in which there is one that binds to the, as I said, to a tumor antigen like EbCAM, and then binds to biotin and on a cell surface. And you can either bind to the antigen positive or to the lipid, and you get, again, modification of the cell surface. Now, one of the issues with hydrophobic and surface techniques, and they've been tried for actually a number of years, almost all of them have to do with lipid interactions that interact or intercalate into a membrane, usually on a single, maybe a double, but usually it's just with one lipid. And these tend to be hydrophobic insertion. They tend to be not very stable interactions. They only last on the surface for maybe a few hours at most. Why this is so attractive is it can be very fast. I forgot to tell you that those T cells that we modified with those rings earlier, they modify the T cells within minutes. Within 30 minutes, it's all gone. And so instead of taking 6 weeks to make a modified cell, we're talking 30 minutes, just moving it around 30 minutes and you're ready to go. For these rings, what we thought is because we have multivalency potentially of the lipid interactions on the surface, that we might now solve this problem of having a greater stability on the surface of the cell. And it turns out what we do, that when we look at the stability of these lipid anchored nanorings on the surface, that they're there for days, up to 3 days. We don't see them leave. And in this case, this is the lipid itself. And the lipid over time basically gets transferred, either internalized or it can be transferred to another cell. If we look at cell-cell interaction, because the rings are in lipids, they might be transferred to another cell. They might sort of hop over to another cell. What we find is that in that case, it's a little hard to see here, but there's a gray here. That would be the amount that might occur, and there's almost none of ways that occurs. We do see some loss of the lipid for those parts, but essentially, we don't see any transfer, almost no transfer of the rings from one cell to another. So they're stable in the cells and with this avidity effect. And so here's cells that have no CSANs. These are target cells, these 231 cells. We then take some cells, these B-cell leukemia, and we modify those cells with our rings, with the lipid and the rings, and we can see here the green ones, and you can see they're all modified. They're forming cell-cell interactions and forming and interacting with the cell that they've targeted to. We can add trimethoprim and we can wipe them from basically the surface, that surface. How long does that take? Well, it can take with this just 1 hour. Essentially, we can remove those. Probably actually maybe even faster. But for the emphasis of this experiment, we can see that they're pretty much within an hour. We can remove them from the surface of the cell. If we've even tried to put 2 or 3 cells together, in which if you use say a DPCO where you have the click chemistry on one cell and you use a biotin on another cell, and they bind to actually 2 different antigens, they can actually target and another cell, and so you can get 3. You can have basically, in this case, you've got multiple cells that are targeted to the same cell, and so you can get, depending on how you do it, you can actually engineer multiple cell-cell interactions in this way. Now, lastly, so we're using those. So we were using, there's still components here. There's some complexity with this. And one of the things I've learned in translating something into the clinic is you want to keep reducing complexity if you can. For a number of reasons, but you want things to be as simple as possible. The number of parts that you have, if you can reduce that number, great. And so you want to see how far you can get things down to be as simple as possible. And so we have multiple components here with what I just showed you. We have to modify the lipid, the cells with these lipids, et cetera, and have to do this chemistry. So what we thought was, what if we could put the lipid on the ring first? In other words, that we could produce it with the ring, with the lipid on there. What lipid would you get? So with my colleague over in chemistry, Mark DiStefano, we had this conversation. I knew he was working with farnesylation, or the lipidation of proteins. He's an expert in that. And what we reasoned is that if we could put one of these signals on our protein or monomer, then we could enzymatically modify that ring with a lipid that is referred to as farnesyl prenylation. And there's 2 types of those lipids, farnesyl and geranyl. These are natural lipids that are used in cell signaling a lot, and they're used to hold proteins into membranes. And so we thought we could use these as so that we can modify the rings, and we wouldn't have to actually modify the cells with these other lipids. And so the idea is that the monomer, you then make your ring, add your bismethyltrexate, make your ring, and then add it to the cell. It goes right into the cell. It has a multivalent interaction, so therefore would be very stable. And then that will then drive, you'll get your cell-cell interaction because you labeled this with a ligand, with a protein that will associate with target the other cell. So we made those, and here's some. These are. I want to point out that just a quick example that farnesyl is shorter, and it has 15 carbons, whereas geranyl has 20 carbons, and this allows us to look at the size or the length of the lipid that we're approaching to see if there is an effect. So these are actually some cryo-TEM images of our rings. that have either that have been either non-lipidated or lipidated with the farnesyl or general, and the rings actually form quite well, quite nicely. We then look to see how well they bound to cells. Again, this is our flow experiment again, and we can see that the rings bind not only to the B cells, they bind to the lung cancer cells, and they bind to the breast cancer cells. So they seem to be agnostic with regard to the type of cells that they bind. Although there appears to be a little bit of difference between them, they appear to be able to bind to essentially most cells, any cell that we're currently looking at. This is just some confocal to show you that, in fact, at certain levels, we've seen that the lipidated cells will bind, the farnesylated cells and germinated cells will actually bind into something called lipid wraps. And these are concentrations or domains within the membrane that are segregated from the rest of the membrane that have a lot of cholesterol in them, as well as other proteins. And they're referred to as lipid wraps. And they're quite fascinating. And we knew that actually farnesyl-based phenylated proteins will have a preference for lipid wraps. And sure enough, what we see is that you can see by the overlap, this is the C-strands that's labeled with the rings have been labeled. The lipid wraps have been labeled with red, and when you merge them, you get the yellow color because green and red go to yellow, and you can see you've got they merge, or they seem to be associated with those same domains. So one thing about this system is it's quite modular, and we're taking advantage of that, and so that we can modulate the number of potential lipids on the ring by having a lipidated one and a non-lipidated one. And so we can, if we just change the ratio, we would have either 1-to-1, either, for example, if we have this, it'd be 4. If we have all of this one, it would be 8 rings. If we have none, it would be 0. So from 0 to 8 lipids could be on the ring. And what we found is that, and this is of course on average, is that for the farnesyl, if we look at the dissociation constants from the cells, what we find is that indeed the farnesyl binds less tightly than the generation if you have just two lipids on average. But as you increase that down to 4, down to 6, down to 8, that kind of vanishes. And so you do get an advantage of generation. It seems to stick to the cells and encapsulate, impregnate, intercalate into cell membranes a lot better. But as you increase that in number because of avidity, the farnesyl catches up basically with the general. If we look at the stability on the surface of the cells, what we find is that, interesting enough, that we do start seeing a difference out to days. And so this is the solid bars. The orange is the general. The blue is the farnesyl. And what we see is that pretty much the same, that we see basically like 80% of the rings are still there after 2 to 3 days. But as we go out, we can start to see actually a significant decrease in the amount of farnesyl. So it looks like you still see some difference if you go out far enough. And these are just the monomers, and this sort of tracks with the monomers. The monomers bind less well, but the generate monomer binds better to the cell surface than the farnesyl. This shows you in the farnesyl that we can add trimethoprim and remove those. It goes down to basically we detect monomer, and so we can get rid of the rings, that ring formation on the surface of the cell quite quickly. This shows you, this is some MCF7 cells, another breast cancer cell line. When we add the farnesylated cells, so these would be Raj cells that have been farnesylated, that have C-strands on them targeting EbChem on the MCF7 cells. These are modified cells. You can see they're associating with the tumor cells. And in a generation, we see it too. We can actually see it looks almost like there's a bit more that's associated with the generation. If we add trimethoprim, we can wipe them off the surface of the cells. We can disassemble them from the surface. Does this matter in terms of activity? So the last slide is that it turns out, so we wanted to look at, can we answer a biological question? One of those is T-cell targeted killing. Is there a time frame? Because if we use a trimethoprim, we can stop potentially that cytotoxicity activity or activation, and does it matter? When are the cells pretty committed to that? So we took T cells and we found that we can disassemble them from the surface of the cells actually quite quickly, within about 10 minutes. Actually, almost all the rings are gone from the cell. And it turns out that when you do that, you do affect cytotoxicity. So in fact, here's under these conditions, the cytotoxicity. I add trimethoprim, and I can essentially remove. Essentially, the cells are at baseline in terms of cell killing. Now, if I follow this with time, in which I'm going to add trimethoprim at various discrete points in time, what we find is that by hour 2, the T cells basically the killing that you get from that's been imparted to those tumor cells is essentially maximized, and you can't go back essentially. And so up to 2 hours, though, there you can actually revert and stop that from occurring. So there's a window of time that you have, and so that was interesting. Nobody had actually identified when that point is. Now this is a modification. This is a modified T cell. It's not like a normal T cell. So under this set of conditions, we can see we can get rapid inside of cell killing, and then that commitment to cell killing occurs after 2 hours. The cells are committed to killing. Now we've gone on and we're using these for a variety of other things now, starting to use these. For example, we're very interested in stem cell modifications for a variety of different applications, particularly for regenerative diseases. And so we're looking at how, with colleagues, to look at how we might be able to help in associating stem cells better and more productively with tissues that for either genetic diseases or for tissue regeneration. So we're starting to branch out into other things besides cancer. So what I thought I've shown you is that we've got this kind of with these nanorings, we've been able to come up with what we can like a little Lego toolkit in which we can have different types of ligands or different types of chemical dimerizers and lipids and ligands that can be mixed and matched with the C-SANS, basically Legos, that allow us to build whatever kind of rings we want, that have capabilities that we want, to allow us to then tune and adjust the cell-cell interactions, that we're interested in. Now, I don't do this work, I of course, as everybody always says, but I want to particularly point out Jake Peterson, who did all the work with the EbCam and CD133 work. He is now Dr. P. And Cliff Sizemore, who did all the work with the lipidation work that I showed you with the biotinylation work and the click chemistry work, and then Yao Wang, who I don't think I have actually, but here's a picture of him right here, who's done all the work with the farnesylation and the prenylation work that I just talked about. So that's Yao, and here's Jake, and then the MD PhD student Cliff, who's now at the Mayo Clinic. Okay, I'll be happy to take any questions. Thank you.
Thank you very much, Rick. That was really remarkable work. Looking for questions either in the chat or the Q&A box, but I'd like to harken back to that plot you showed of where your modified T cells end up and how many of them end up where you want them to, and then compare that with some of the similar plots we saw for nanoparticles yesterday. Mm-hmm. You are maybe 5 orders of magnitude better getting nanoparticles in place. Right. So it's a really remarkable, kind of key and lock approach here. As I was listening, I was wondering how durable those nanorings are in attachment to the T cells, and you kind of indicated that you can turn them on and off at will, right?
Right. And yeah, so when we're thinking about this clinically, how we're going to use this, is that there's sort of 2 switches. One is that when you give a dose, they will go and bind to the T cells. They will see there's, and we show that in animals. And then there's a stability there on the T cells for several days, those T cells. But you really don't want the T cells modified forever. Okay, and so what happens is those T cells engage. When they engage with the tumor, they divide, they proliferate, they start killing, and they make other ones. And in that process, each time they divide, you got 50% less, 50%, right? You're diluting the amount that's on the cells after you've given your dose. And so that sort of tunes down the T cell activity until you give another dose. And so that's one mechanism for control of the activity. The other one is using the trimethoprim, the drug, that can go and lead to rapid dissociation of the rings. And that allows you to have the ability to temporarily do that, to temporarily go in and stop that interaction, the possibility of the interaction between the T cells and the tumor cell, or wherever you want it to go. And that's important because the most common side effect of these kinds of therapies is what is referred to as cytokine storm. And in fact, that coronavirus, a number of patients who do very badly, they have this overactivated immune response. And that activated immune response becomes systemic, and it basically can kill you. And so that's the main worry in any of these sort of immunotherapies too, and is the main toxicity. And so this allows us to have a very easy, very cheap too, I might add. It's off generic, basically antibiotic that allows us to do that. Current methods for addressing these toxicities are very expensive because they involve antibodies and other things that have to be given to the patient. And they're actually quite expensive, tens of thousands of dollars, per dose. So that's one of the kind of advantages that we have.
Indeed. I was also impressed at how quickly and efficiently the nanorings found the T cells themselves, and that is just a function of circulatory.
Right. I think it's just mass action, and that the what we go for is there's T cells that are in circulation. There's T cells that are in the various lymphoid tissues, like the spleen, the lymphocytes, particularly the spleen, because in this case the spleen is what filters the blood. And there's T cells also in the liver. But what happens is what cells are you going to see first? And you're going to see those first before you actually get circulated and get to the tumor. And so if they hit the tumor first, it's not a bad thing because T cells that wander into there are going to recognize that and will kill the tumor too. And so we actually have we can tune this in which we have more targeting ligands on the ring for the tumor and fewer for the T cell, so that the majority of it would go to the tumor first, and then the T cells would find it. Or we can tune it where it would go to the T cells, and the T cells would then find the tumor. And so we have this kind of flexibility to do that. And in some cases, you may want one or the other, depending on what you're thinking about how you can do it, because we can just mix and match basically on the ratios.
Very powerful platform, and
thank you.
Great work with it.